everyone. I'm Guilin Tang from MD Anderson. I'm a cytogenetics and hematopathologist. And today the topic is a optical genome mapping in T-lymphoblastic leukemia, also called a TALL. So I have no conflict of interest. TALL, it is a neoplasm of lymphoblast committed to the T cell lineage. It accounts about 15% of PD-AL cases and then 25% about the adult AL cases. So TAL historically was classified into immunophenotypical subtypes. It's based on the T cell differentiation. So called as a pro-T, pre-T, cortical T, and then medullary T. So currently the pro-T and then pre-T are lumped together into an earlier T precursor, so it's a ETP-ALL. So I'm going to tell you why it is so important of the ETP later. So under the current WHO classification, just classified as a ETP versus non-ETP. On the international consensus of the classification, that has a the three big category. So ETP with BCL11B rearrangement, and then ETP NOS, TALL NOS. And but under this category, there is a proposed 8 preventional entities based on the cytogenomic aberrations. So this is a figure shows you the immunophenotypic features, the different stage, and then also the correlation about the cytogenomic, the abnormalities. So on the first stage, it is a ETP. So this is the stage, account for about 15% of the PDTL cases, and then 20 to 40% of the adult TL cases. So this is a very early stage that still express the myeloid markers like the CD13, CD33, CD117. The big one, so usually has a kind of a challenge, to differentiate with a mixed phenotype of acute leukemia. So has a myeloid and then T lineage, the components. So also you see, so the Notch1 mutation and then the CDKN2A, the deletion, it is very common in the TALL. But in the ETP, the Notch1 mutation is much less common compared to the later stage. And also CDKN2A, the deletion, it is really rare in the ETP stage. So this kind of shows you on the early cortical or the later cortical, the major pathways involved. So as I said, so at the ICC classification, BCL11B activation, it is a real entity, and then there's also there's 8 the prevention entities. They are the T cell lineage transcription factors. So this one gives you the frequency, how often it is, and also here it shows you the partner genes. So this one thing is very important, so it is like the BCL11B. So so it is the BCL11B not only has a category for the activation, sometimes and then there's also has a the other partner genes to activate the other pathway. So I'm going to talk a little bit more later. So this is the one that shows you the distribution of the main genetics subgroups in the TALL. The top one is adult, and then the lower one is a PD group. So it is really based on the T lineage transcription factors. And then there has a slightly difference, like for example, the TLX1, TLX3, So this is one, it is like a TL3 is more common in the PD group, and then TX1 is more common in the adult group. So as I said, BCL11B now is a really, really hot topic in the TALL. So but it is not easy for us to detect. So first of all, it has multiple partner genes, and then also under the different break points. And then some kind of the like a break points, so involve this translocation is far more like a symptomatic part of this gene. So make the fish also the fish probe design is very difficult. And also because this one it is on the chromosome 14q32, it is really on the terminal. So sometimes conventional cytogenetics also difficult to detect this rearrangement. So for this part, so the OGN, I think it has a great volume in the detection of the BCL11B, the rearrangement. So this is, I give you one case. So this case shows the 8-14 translocation. And then the break points is following the CCDC 26 on the chromosome 8, and also the BCL11B, but it is a kind of a little bit off of the break points. But this is a kind of rearrangement. It has been confirmed the activated of the BCL11B. So now I'm going to talk about the comparison between the OGN and our conventional cytogenetics in the TAL, our NAB, the experience. So the first one, it is a pilot study. We sent out for the Bionano back to the 2021. So we total sent out 8 cases. So 4 of them has an abnormal karyotype and 4 with a normal karyotype. So but so on the right side, I list the OGN, the result, the copy number change, and then the rearrangement. As you can see, all cases has at least 2 or more, the cytogenetic abnormalities. And we do see many of them is recurrent, like the deletion 9p, the 17p deletion, 17q deletion. And also we see a recurrent, the chromosome rearrangement. So I give you one example. So this case showed a normal diploid karyotype by conventional cytogenetics, but the OGN detect multiple abnormalities, including the 1011. This is a recurrent cytogenetics abnormality, and also there's a couple of the numerical changes. So about the 1316, this is there's a fusion gene. It is not reported before, so we don't know the clinical significance. And also, of course, it has a 17p deletion. So this one, it is also a high risk. So on this kind of the pilot cohort, we detect all the cytogenetic abnormalities in all cases. And then a couple of them are recurrent, like as I said, CDKN2A deletion, the 17p deletion, and then deletion 13q deletion. And we also see the these are the cases that has been reported before is a recurrent. And then of course BCL11B the rearrangement, this is one, it is a very important one. So, and then the 1011 also is very well studied in the AML, but it's less studied in the TAL. So now we kind of we have expanded our study to 28 cases. So the first one I just show you, it is a kind of overall impression. So we had 16 cases showed abnormal karyotype. 11 cases show the normal karyotype, and 1 case is insufficient. So there's no fusion gene detected by the chromosome analysis. By the OGN, so we detect the cytogenetic abnormalities in all cases. And most importantly, we had a fusion gene detected in the 18 cases. That is accounted for the 64% of the cases. So we kind of subsequently to divide it into different group. Like for the completely coordinates means all the chromosome abnormalities are matched between the karyotyping and the OGN. And then there's another case is a coordinates where the fusion gene means abnormality picked by both assay, but our OGN picked up the fusion genes. And then for the partially coordinates, this account for half of the cases. So means within the same case, some abnormalities detected by both assay, but some abnormalities only detected by the OGN. And then these 12 cases, it is a cases has a abnormal normal karyotype or insufficient karyotype by the conventional cytogenetics, but we detect abnormalities by the OGM. So this is one we put together. So OGM detect additional information in the 96% of the cases. And then the clinical relevant one is detected in the 57% of the cases. So now at MD Anderson, we have done more than 500 cases. And then we found, so TALL had a kind of most dramatic or the findings by the OGM. It is very, very valuable by the OGM in the TAL cases. So this is one I'm going to show you about the fusion genes. So we had 18 cases, so the fusion gene, and then 15 are the recurrent. So the first one, it is show you we have 4 cases, so the translocation to lead to the BCL11B activation. So this is usually it is a worst prognosis. And then we had 4 cases also shows involve the TLX1 or the TLX3. So this is one category also. So and then we also had 5 cases. It is belongs to the category of the HOX-A. So it is I present the cases 1011 translocation, 1119. So this involves the KMT2A gene. And also has one cases has an interstitial small deletion on the 9q34, so leading to the genes fusion. So this is one is again belongs to the HOX-A category. We also had 2 cases involve the CCND2, the one. So this is why it is involving the cell cycling. So the CCND2, very interesting. Also, we detected in the TAL before. We never see the CCND2 in the TAL. So, and then we also have 3 cases showed the novel fusion genes. So this is a category also very interesting. I mean, for the further study to see what is the function. So here I just show you the numerical change in the all the 28 cases. Most of the cases show at least one of the numerical change. So also there's a kind of the aneuploidies, right? So monosomy and then trisomies. So I didn't count in here. So this is one is really count for the deletion or the gain. So interestingly, you see the CDKN2A deletion we detected in the patients. But as you see, in the ETP, the newly diagnosed one, there's none of the cases so that the 9P deletion. Another interesting finding, it is a IKZF1 deletion. So this one is very well established in the BLL. So it's associated with a poor prognosis, but it is not well studied in the TALL. I think it is also maybe when we have more cases as a interesting area. So in summary, we had 57 the cases showed abnormal karyotype by conventional cytogenetics, and then about all the cases showed chromosome abnormalities by the OGM. So all the abnormalities detected by the conventional cytogenetics all picked by the OGM. So the sensitivity is 100%. And then most importantly, OGM provide additional information in the 97% of the cases, and then 56% of the cases had the information is clinically relevant. So and then also most importantly is detect the fusion genes. So in more than half of the cases. So so 15 were the recurrent. So the discovery of this the recurrent the fusion genes could improve the TL subclassification, and then 3 were novel. So that is all my talk. I would like to thanks for our MD Anderson's cytogenetics team, and also we have the very brilliant technologists help us to develop this assay in the lab. And I also would like to thank for the Bionano team for the strong support during all our the development of the OGN. Thank you.